Hello, everyone. This is Dean Malvick from the University of Minnesota. I'm a plant pathologist based in St. Paul. I've been working on soybean and corn diseases for a while, and sudden death syndrome, the topic of today's presentation, is one of those diseases. And so I'm going to start with a very brief background to help you understand this disease that's been spreading across the region. So sudden death syndrome, we'll call SDS for the rest of this presentation, is caused by this soil-borne fungus called Fusarium virguliforme. Now, many of you know that Fusarium is a common pathogen on many different crops. This one's particularly unique for soybean and some other legumes as well. So yield loss on soybean can exceed 50%. So this is a very significant disease when the weather conditions are favorable. But usually that disease loss isn't that great over entire fields because the disease tends to be patchy. But occasionally it can be widespread over entire fields and cause a lot of loss. So the yield loss depends on when plants are infected, early versus late, the extent of the problem in the field, the weather, and the soybean variety. And we'll talk more about this as we go along. So SDS, unfortunately, is often severe in well-managed soybean fields with a high yield potential. So we can often have a very good-looking crop throughout the summer, and then August start looking very bad because of SDS. The pathogen also causes disease on dry edible bean, and I'll explain this more in a few minutes. So it's not just a pathogen disease of soybean, but dry edible bean as well. This is not a new disease, and we'll talk more about where it's been. But here's an example from 20 years ago in Illinois. There was an estimate of over $400 million of loss across the state. And now SDS has been there much longer than Minnesota and North Dakota, and it's spread across the entire state. But again, to indicate when the weather conditions are suitable, this is a disease that's very significant. So usually, as I mentioned, SDS, being a soil-borne pathogen, starts and damages the crop mostly in patches in the field. Now, these may be the size of a pickup truck or acres in size, and they spread over time. Here's an example. This is a field in southern Minnesota, not far from Owatonna, near where SDS was first found in Minnesota. But there's a part of the field here, as you can see in the circle, that's where the disease was first found. And we watched this disease over the years. That's where it first appears in the field. And then as we have the right weather conditions, it has spread across the field over the years, so it can cause a problem over the entire area. Now, how much yield loss can this cause? I mentioned 50%, and that is in the infected patches. Again, uniform infection across large fields is not real common, but here's an example of a field trial I put out a few years ago in a field that did not have a background population of this pathogen. Some of the plots I added the pathogen to, some I did not. And you can see on the right, the susceptible variety yielded 45 bushels per acre where there was SDS, and where there was no SDS, it yielded 65. So we lost 20 bushels an acre in this example. And it wasn't based on seed size, it was based on seed number per plant. SDS is spreading, which is a key topic for us in the northern area here especially. If we look backwards, back to the early 1970s, it was first found in Arkansas. And you can see the dates, moving up from there. 79, it was first found in Illinois, 93 in Iowa, 2002 in southern Minnesota, and then 2018, it was confirmed in North Dakota. So where is it in our states here, Minnesota and North Dakota? Again, the circled area with the red in the bottom shows where SDS was first found. And now it's spread pretty widely over the bottom, third to half of the state. Now it's certainly not in every field by any means. We haven't even confirmed it in every county. You see some white counties, and that doesn't mean it's not there. We just haven't gotten a sample and confirmed it. But you can see the progression and there are dates here. It's definitely moving north and west. And we've confirmed a few more counties in the past couple of years. So this is what we've expected based on what we know about it. Against the soil-borne fungus, anything that moves infected soil or infested soil, I should say, can move this pathogen around. And what about North Dakota? As I mentioned before, it was first found in the very southeastern corner in 2018. It's been found in more fields than it was then in 2020. And this year it was also confirmed on the far northern border of North Dakota. So it has been a spreading disease there too. So you may look at this pattern as we do in Minnesota, and we've seen this before. It looks like there's a big jump, and maybe it is. But chances are there are fields in between these two locations that are infected too and just haven't been reported. And I suspect there's more infection in northwestern Minnesota too than we have confirmed at this point in time. So how do we know where SDS is? You know, scouting for it, usually the symptoms start appearing first in early August. Sometimes we don't see them until mid-August or even later August, you know, depending on the weather conditions, etc. Where SDS is often, not always, first shows up in low, poorly drained or compacted areas of fields. And the pattern isn't always that case, but those would be the place to look for it first. And what to look for yellow and brown patches between veins on leaves. See a little bit on this picture here, but this shows it much more clearly in these larger pictures. If you look on the picture on the left, you see a trifoliate of soybean. You see the SDS are actually different stages of development on that single trifoliate. And so you can see the one in the lower on the right in that picture, the early yellow chlorotic patches developing. And this is a result of a toxin. And you can see the top center one, a little bit more advanced, a little bit of brown necrosis developing, and that other trifoliate on the lower left, a little bit more advanced. If you look at the larger picture, then on the right, you can see more complete symptoms as it's developed further with lots of chlorosis, that brown dead tissue, the necrosis between the veins, and the leaves tending to curl in a cup as well. And so the symptoms result from two things. The disease infects the roots, causes root rot, as shown here. And sometimes under wet conditions, we can see this blue coloration of the fungus growing on the root surface, although we don't always see that. So that's not an absolute diagnostic kind of feature, but it is something to look for. And as that fungus grows in the root tissue, it also produces a toxin that then moves up the vascular system of the plant to the leaves and damages the leaf tissue, resulting in the symptoms that you saw on the previous slide on the leaves. Other diseases that can be confused and are often confused, I should say, with SDS. Probably the most common in Minnesota and our region would be brown stem rot in terms of the leaf symptoms. The root rots caused by the SDS pathogen, Fusarium, can look very similar to those caused by other fusarium pathogens, such as Fusarium solani, which is widespread across our region. And there's other diseases that can cause leaf discoloration that can look somewhat like SDS as well. But again, probably the most common mistaken disease in many areas is brown stem rot, another fungal disease that is widespread across much of Minnesota. And I believe it's in North Dakota too, although I don't think we have a full understanding how far and wide it's spread there. So again, brown stem rot on the right, SDS on the left. Sorry, my slide advanced automatically there. You can see the leaf symptoms look very similar. So if we split the stems, you can see the brown stem rot has this brown discoloration in the pith, and as you can see in the lower right picture. The pith of the SDS plants do not have discoloration. Although if we go just under the epidermis, the skin of the lower stem, you can see this sort of gray-brown discoloration. And that's not in the center in the pith, that's just under the outer part of the stem. Those are distinguishing features for brown stem rot versus SDS. And again, looking at two sets of leaves from a field. This happens to be from the same field. SDS symptoms on the right, brown stem rot on the left. You can see more brown discoloration there, less chlorosis, but it's not always even this distinct. So again, they can look quite similar, and it's important to tell them apart because we manage them differently. Also, as I mentioned before, the pathogen, the Fusarium that causes SDS, can also cause considerable root rot and damage to dry edible bean. Here's an example of pinto bean. On the left, far left, is a picture of healthy plants, no pathogen, and the B shows in our field plots where the pathogen, the disease is. It's stunting the plants, causing this sort of this sort of general chlorosis on the leaves and a little bit of necrosis. Doesn't cause the distinct symptoms on leaves of beans as it does on soybean. And then it causes root rot, as you can see on the far right, very similar to fusarium root rot on beans. So what are some of the risk factors associated with this disease? First, field history of SDS and the presence of the pathogen, obviously. And wherever the disease was before, it tends to occur again. And the presence of the pathogen, it's hard to know where that is until we've seen the disease, but it's probably spread further than we know at this point in time. Compacted soil, poor drainage favors it, high soybean cyst nematode, and of course, susceptible soybean varieties, since we do have varieties that have high levels of resistance now to SDS in many areas. Wet soil 2 to 3 weeks after planting, especially after that seed germinates. That's when the initial infection occurs, and then moist to wet soil throughout the summer. So again, the disease tends to occur in hot spots or patches in fields. So a few years ago, we did a study in Minnesota and a few other states trying to see what is distinguishing these hot spots, which tend to be consistent to some extent from year to year. Although again, the disease does spread across fields. What distinguishes them? So we went on 11 fields in Minnesota and 11 fields each in Illinois and Iowa. And we sampled from these hot spots where SDS was prevalent. And then we went beyond those spots where there were no SDS symptoms. And we sampled the soil. We did complete chemical analysis of the soil, organic matter, and other soil properties. We looked at soybean cyst nematodes, and we look at microbes in soil. So I want to mention that microbes were found to likely have an association with this. Other microbes in the soil may be playing a role in where SDS develops. Now, there's been a lot of talk about SCN interacting with SDS. Again, SCN being soybean cyst nematode. And there definitely is a relationship. High levels of SDS are often associated with high levels of the soybean cyst nematode in the soil too. It's not an absolute. We don't need high levels of SCN to get SDS, but it tends to be more severe. So we asked the question, do we see different levels of populations in these hot spots versus the cold spots in these fields with SDS? So in these 11 fields across Minnesota, the red bar shows the hot spots, the number of nematodes, the eggs per 100 cc's of soil, and the blue bar shows the cold spots. You can see there's no absolute clear pattern of which have more soybean cyst nematodes. We see some of the fields where the hot spots are higher, some there's no difference, and some the cold spots are higher. So we didn't see a clear relationship there. If we average it and put all that data together, we see the SCN counts for the cold spots and the hot spots on the average being very similar. Again, being different across those locations. And so there isn't a clear relationship there. But it could be that in all of these cases, we had enough nematodes to help enhance SDS. So how do we manage this disease if and when it comes into an area that we're trying to manage? Number one is know the fields of greatest risk. So that comes back to scouting. Some of the management options are expensive. We don't want to use them very likely unless there's a high risk. And so we need to know where the risk is highest. We can use resistant varieties, and that is our best option if they're available with the other trait package that we want. And then we can use specific seed treatments, and I'll give examples of all of these. Seed treatments, we have 2 fungicides that are very effective, Olivo, now from BASF, and Celtero from Syngenta. We also have a biological seed treatment called Heads Up that comes out of Plant Protectants in Saskatchewan. So moving into SDS. So here, again, we have in many areas, we have many varieties that have moderate to high levels of resistance to SDS. None of them are completely resistant, but it can make a big difference if it's resistant or not in terms of how it performs in a field with SDS. And again, this is the first choice, I would say, to manage SDS if we can find a variety that matches our needs. And here's an example. Two varieties, MR, if you look across the bottom, it says MR and MS, that's moderately resistant, moderately susceptible. Okay, in a field with SDS, the moderately resistant one yielded 56 versus moderately susceptible was 45 bushels an acre. So there's an 11 bushel difference just by planting a different variety. With that moderately resistant variety though, when it was planted in a part of the field with no SDS, it yielded another 10 bushels. So again, even the moderately resistant variety was losing to SDS, losing yield, but certainly was performing much better than the more susceptible variety. So the other topic here, of course, is seed treatments. If we don't have a variety or can't get a variety that has good levels of resistance to SDS, or we may want a field where we want to combine the two for even more protection, we have a few seed treatments, as I've already mentioned. For a while, we've had Olivo for about 6 or 7 years. Fungicide, again, from BASF. And then more recently, we have Saltro. And I'll mention some results from both of those. So first of all, Olivo's been around the longest. It was the first seed treatment product that was highly effective against SDS. And I'll mention 2 studies. Here's a simple comparison that I did in Rosemont, Minnesota a few years ago. I have a variety that was fairly susceptible with Olivo and with no Olivo. And this is at the higher labeled rate, the common rate recommended. And you can see the disease severity or disease factor, disease index, we call it the DX, was 51 versus 14. As you can see the variety X in the first column. So we dramatically reduced the amount of disease with the product on the seed. And you can see the yield, we increased it from 61 to 73 bushels. So again, 12 bushels just for the use of that seed treatment. So again, it showed a lot of efficacy against SDS. And I'll just mention in a multi-state study across the region in multiple states, including Ontario, where Olivo was tested with the low and high rates, both of them, the rates reduced root rot and SDS foliar disease and increased yield compared to the base treatment. Again, so this product has been shown to widely be effective against SDS. Now, again, as of just a little over a year and a half ago, Saltro from Syngenta was registered for SDS management as a seed treatment. And so now with these 3 products on the market, there was very little data available for Saltro. So I set up a study in Rosemont and Waseca, Minnesota. I put it in plots that were inoculated with the pathogen and they were irrigated to help enhance the disease development throughout the summer. We had 4 treatments: untreated seed, a base treatment, which was Acceleron, or Acceleron plus Olivo, Acceleron plus Saltro, or Acceleron plus Heads Up. I had both a moderately susceptible and a highly resistant variety in those plots. And so first of all, I'm gonna focus much of what I show you here on Rosemont, since we had better results from there. And the study was not confounded by brown stem rot, which developed in our Waseca plots. So of course, we planted this in early May, and we watched the plots and the plants grow throughout the season, showing, of course, as typical, very few symptoms until August. And so in late August, the disease really started to develop. And here's an aerial image taken. You can see the yellowing on the plots, and that's all due to SDS. And the darker green plots, you can see have a high level of SDS management. And those happen to be the plots that primarily have the Olivo and the Saltro. And again, you can see, say, treatment 5 and treatment 4, you can see 2 of them side by side right in here. And treatment 9 and 10, again, Olivo versus Saltro. You can see those patterned down and in the other blocks with the same treatment below. So this is a study with 11 treatments replicated 4 times across the field in these blocks from upper to lower part of the picture. Again, the dark green areas showing a high level of SDS management with those treatments. And if we look at the treatment with heads up across, we can see also it tends to be a little bit more green than the base treatments as well. If we look at the actual SDS disease severity across those locations, what we have here are the resistant or the moderately susceptible variety is the blue bar, excuse me. And then the more resistant variety is the red bar. So this shows the disease index, again, on a 0 to 100 scale. So if we average across these treatments or the plots, we didn't have extremely high disease severity here. But you see a very clear pattern. On the far left is no seed treatment. A base seed treatment, we see a little bit of advantage, surprisingly, with the moderately resistant variety. But when we go to the heads up and the Olivo, going from left to right, and the Saltro at the very end on the right, you can see a clear increase in control of SDS, resulting in lower and lower disease severity on both the moderately susceptible and moderate resistant variety. If we look at this for actual yield, these are ranked essentially from the highest to the lowest yielding treatment. Again, the AG21X9 is the more susceptible variety. The S20JX5 is the more resistant one. So again, you can see on the very last part of the description, so again, it's the variety plus the acceleron and then plus the Saltro. So Saltro had the highest yields in these plots. The Olivo was just behind that. Some of it significantly different, some of it not different. And then we look at the heads up was down just a bit below that, but higher than the no seed treatment generally. So again, you can see how these work. We have some very effective seed treatments now available for SDS management. So key final points. SDS continues to spread. It's not everywhere, but again, we don't know where it is until we see it and we get the weather conditions that essentially brings it out or results in it to develop. Keep looking. It's probably spread more than we think, but again, it's not everywhere. Scouting and diagnosis and being sure what we're dealing with are critical. It's important, especially in new areas, to send plants into a diagnostic lab to make sure it's SDS and not something else. And we can manage SDS with resistant varieties. And partial resistance is available widely in many areas. We're also more limited as we go further north in what we have available. Or we can manage with selected seed treatments. Again, I've shown several that are shown to be quite effective. And if you have any final questions or comments, I think we have a few minutes now. Otherwise, you can contact me